We have all probably seen pictures of vertical farms before, big constructions of rows of purple LEDs and plants stacked on top of one another. But have you ever wondered why purple lights? What if you were to grow plants under blue light or red light? We did exactly that. We studied the effects of red and blue LED lamps on the growth of basil. So why basil, you ask? Well, this hasn't been studied specifically for basil before. Scientists have however studied this with other plants, such as orchids, grapes, and watermelon. Based on that research, we are expecting red light to result in increased growth compared to normal lamps, and blue light to result in impaired growth. So how did we do it? The experimental setup is basically 21 plants that were divided into 3 groups of 7. 2 of those groups were the treatment groups and 1 was the control group. Now we started out with planting a ton of basil seeds, more than we needed. For about 4 weeks we let them germinate and grow together, all in one Tupperware container until we had these cute little plantlets. Then we selected 21 plants of equal size and potted them in their own individual cups. After which we divided them into 3 groups for the treatments and let them grow for 3 weeks. For the treatment groups we used a red LED lamp and a blue LED lamp. For the control group we used a standard fluorescent lamp. After treatments we measured several growth indicators such as shoot height, leaf length, weight, and leaf area index, which is the leaf area divided by the ground area that the plant takes up. This was then statistically analyzed.
Now for the next part we will look at the results of our experiment. After the 3 weeks the plants were taken from their cups and measured with the criteria that we determined beforehand. What we see in front of us is a general overview of all the measured data that we have. Because this can be a bit overwhelming, we will look at some of the graphs that we made. So here we see the first graph. In this graph, the 3 wavelengths are presented on the x-axis of the graph, and on the y-axis, we see the mean shoot height in centimeters. The yellow bar represents light of a full spectrum, so this is our control group. The blue and red light groups are represented with the corresponding bars and their colors. The small arrows that you see on the bars indicate the standard errors of the group. Immediately you notice that the blue and red light groups lag behind the control group, which is something we only expected for the blue light group. We do see that the red light group value is lower than the blue light value. Then in the next graph we see the length of the leaves in centimeters. There is a similar pattern as in the previous graph, but we do see that the growth of the leaves has decreased a lot in the blue light group, which is what we expected it to be. And now in the third graph we look at the leaf area index of the 3 groups. We were really happy with this result as we expected increased leaf growth in the red light group. The blue light group once again is the lowest of the 3. Finally, we looked at the fresh weight of the plants. These values were equal on average between the 3 groups. This was a little bit strange to us and it did not help a lot in the analysis, but it was still an interesting thing to note.
In this study, we investigated the effects of different wavelengths of light on the growth of Ocimum basilicum. Our results indicated that growing plants under either red, blue or full spectrum light has a neutral effect on the growth of O. basilicum. However, this does not support our hypothesis as we expected enhanced growth for plants grown under red light and impaired growth under blue light. A few reasons for our hypothesis not being supported by our data could be a sample size that was too small, we only had 7 plants per group, not being able to find out how powerful each lamp was, shortage in space, the groups couldn't be next to each other, and our LIE or leaf area index is inaccurate as we were not able to use the best method to determine this. To improve our experimental setup, we could have a bigger sample size. A similar study uses 552 parsley seeds for a lead experiment, which has a much higher chance for significant differences. And we could also measure the chlorophyll content and root characteristics of the plant.